Welcome back, students. Today we're going to discuss the second chapter of the regulation of gene expression. In this, we'll be going to discuss the gene regulation in prokaryotes. So, under this heading, we'll discuss the basic structure of what an operon is. Now, operon is the method by which prokaryotes regulate the gene expression, and then we'll take up two illustrative examples of the operons, the lac operon and the trip operon. So let's just begin. Now, bacteria present interesting method of gene regulation of gene expression. It is via the form of operons. Now, what is an operon? An operon is a cluster of structural genes. Structural genes are the proteins which are destined to perform certain functions. So it is a cluster of structural genes which share a common promoter and other regulatory sequences. So what is now, this clearly means what is now happening? There will be one common mRNA formed, and this common mRNA will have those multiple 1, 2 or 3 genes together. This leads to the formation of a polycistronic mRNA. Cistron, is the segment of the nucleotide sequence which leads to the formation of one particular gene or protein. So the very mechanism of operon results in the formation of polycistronic mRNA. The first example of the operon that was described, and it is the most illustrative example, it has helped us a lot in making our understanding towards gene regulation and bacteria, was the lac operon. This is a representative diagram of lac operon. You can see that on the right side of the figure, 3 genes are represented by A, B, and C. Now these are the structural genes, and on the left side of the figure, you have the regulatory sequences in the form of activator, binding site, promoter, and operator. We'll be taking this up again in detail in a short while, but for now it's enough here to understand that how the operon is located, what is the structure of it, the structural genes located close together, and all the ones are being controlled, regulated by a single set of regulatory sequences. So what are the basic components of an operon? The basic components of the DNA in operon can be explained as a promoter. A promoter is the nucleotide sequence that to which the RNA polymerase goes and binds, and hence it helps in translation of the gene. Then we have a regulator. A regulator is the sequence where the other regulatory elements which help the RNA polymerase go and bind, the elements being inducers or maybe of it may be of co-repressors, etc. And the next is operator. Now, operator is the sequence, as you know from your previous lecture, to which the DNA repressor binds. In case of lac operon, classically the operator is placed just next to the promoter and between promoter and the structural genes. So you must remember, if we are talking about an operator, and then that means we're talking about a repressor binding site, and then that means that it is an example of negative regulation. Then we have the structural genes. So these are the genes which are being co-regulated, the regulation of whose should be fine-tuned and regulated together in order to achieve the particular effect. Let's now get to lac operon. So lacoperon was the first explained by Frank Jacob and Jack Monod through their works in 1960. Uh, Lacoperon is, I must give you some brief about lacoperon. It is the operon which helps the bacteria utilize lactose as the fuel. Now, lactose is not the primary fuel of the cell. Glucose is the primary fuel of the bacterial cell. So, lacoperon is only active when glucose is not available and lactose is present. So it will only be, it becomes a very beautiful example, as it does not have to remain turned on all the time, and it only has to turn on when some molecular signal is present. So the lac-operon has three genes for the metabolism of the lactose. The first gene being LAC-Z, it codes for the protein beta-galactosidase. Beta-galactosidase cleaves the lactose into its constituents galactose and glucose. So when the cell has to utilize lactose, it has to first break down lactose into the simpler components, and then the simpler components will further be utilized by other metabolic processes. Then the second gene is LAC-Y. LAC-Y codes for the protein lactose permease or galactoside permease. Now, what is this permease? It's basically a transporter for the cell. Now, why is it required? Glucose is not present in the cell, whereas in the environment, lactose is present. Now, this lactose has to first enter the cell in order for the cell to utilize, and then it can only be used. So, to enhance the amount of lactose entering inside the cell, the active lacoperon synthesizes multiple numbers of the permease, the transporter, and these transporters then increase the concentration of lactose inside the cell. The third gene is LAC-A. This codes for the protein thiogalactoside transacetylase. Uh, its functions are not clear, but still there are theories around it. Some researchers believe that it modifies the toxic galactosides within the cell in order to facilitate their removal. Others think that the transacetylase converts lactose into allolactose. Now, we've discussed that the lacoperon is not always on. It has to be turned on, and so for there must be some cellular signal. Lactose in itself is that cellular signal, and it is in the form of its isomeric form allolactose that it acts as the cellular signal. So there must be a method to convert lactose into allolactose so that the lacoperon, the negative regulation of the lacoperon can be removed. So LAC-A does that work. So now the lacoperon relies on the negative regulation. Why on negative regulation? Because it has a repressor on it. It has to be negative. It has to be not turned on all the time, because the cell does not need to uh, exercise extra energy to use a fuel as complicated as lactose when glucose is already present. So it has to be turned off all the time. So there comes the role of the repressor. And you know from your previous lecture that whenever the repressor is there, the operon is said to be negatively regulated. So besides the structural genes of the lacoperon, we also need to familiarize ourselves with other components of the lacoperon before we go ahead and for a further detailed discussion of the operon, of how it actually functions. So the first example is repressor. The repressor of lacoperon is called lac repressor. We know it goes and binds to the operator. The second component is activator. Now, CRP, cyclic AMP receptor protein, or CAP, catabolite gene activator, both names, it has both the names being used alternatively, is the activator for lacoperon. Activator, goes and binds to the RNA polymerase. So and so the then is a co-activator. Now this activator needs another molecule, another protein molecule, regulatory molecule, in the form of cyclic AMP. Now CAMP has to go and bind to CRP, and then this activated CRP goes and binds to the RNA polymerase, which is bound to the promoter, which is just adjacent to the activator. Then they have the operator has an inducer. Now remember, inducer is the molecular signal which tells the in this case the negative of the negative regulation, which goes and tells the repressor to dissociate from the operator and let the transcription go on. So inducer in case of lacoperon is lactose, which goes and binds to the repressor, and or its form allolactose. There is another inducer in case of lacoperon, which is IPTG, isopropyl thiogalactoside. It is a non-metabolizable inducer, and it is frequently used in case of research work. So again, summing up the same thing. So RNA polymerase promoter interaction, for to have this strong interaction, it needs CRP, the activator. The CRP in itself requires CAMP for its activation, right? So the bottom line of this slide is a lac repressor is a negative regulatory element, and it is only responsive to lactose. If lactose is present or not, accordingly, the lac repressor will work. So negative regulatory element, the operon is always turned off. The CRP-CAMP is a positive regulatory element, the complex of these two molecules. How is it? It is positive. It is responsive to the concentration of glucose. So whether glucose is present or not will decide the presence of the active complex of CRP and CAMP. Let's now get to what exactly is happening in the lacoperon. When I mentioned that lacoperon is only turned on in the absence of glucose and the presence of lactose, this calls for explanation of another term called catabolite repression. This method of repression explains the repression of genes which are required for the catabolism of alternate sugars when glucose is present. The alternate sugars being lactose and ribinose, etc. So the lacoperon also gives an example of catabolite repression. So the lacoperon can be understood in 2 or 3 scenarios. The first scenario being when the glucose is present, lactose is absent. In that case, the operon will, of course, be turned off. So what's happening? When glucose is present, there is no cyclic AMP. You must know that already. There is no cyclic AMP, which is a co-activator for the activator CAP or cap binding protein. It is inactive. The activator co-activator complex is not formed, so it will not be able to go and bind to the cap binding site, interact with the RNA polymerase and strengthen its binding to the promoter. Whereas the LAC-I gene, which codes for the LAC repressor, has a constitutive expression. It is constantly being expressed, transcribed, and the repressor molecule is formed. Since there is no lactose present, there is no inducer present, for example, allolactose or lactose, to go and bind the repressor and activate it. So the repressor is in its active form. It goes and binds to the operator, and this in turn ensures that the transcription of lacoperon is blocked and no lacoperon genes are being transcribed. Now, second scenario is when there is no glucose but there is lactose. In that case, the operon will be turned on. In the absence of glucose, cyclic AMP will be formed. It will be available to go and bind to cap and form a functional complex which goes and further binds at the cap binding site, interacting with the RNA polymerase, which is bound to the promoter region. Now, the LAC-I repressor which is being formed will have allolactose, since there is lactose present in the cell, will have allolactose present in the cell, bind to it and turn it inactive by inducing conformational changes. Now, the inactive repressor will no longer be able to go and bind to the operator, hence not being able to block the transcription of the LAC operon. The transcription goes on and mRNA, a polycistronic mRNA, is being formed. The third scenario is when both glucose and lactose are present. In that scenario also the operon is off. Why so? Because when the glucose is present there is no CMP, so cap, the activator, is not in a functional form, it's not active. So it can no longer go and bind to the cap binding site and strengthen the binding of RNA polymerase to the promoter. So the LAC repressor is being formed, and there is, although allolactose present in the cell, which should be able to block the binding of the repressor to the operator, but it is not enough to block all the repressor from binding to the operator. In turn, there is also not enough strong binding of the RNA polymerase, interaction of the polymerase to the promoter, which should ensure the transcription, and hence the transcription is blocked. So what is the conclusion of all this? The combined effect of glucose and lactose on the LAC operon means what? So when lactose is low, the repressor is bound. This means it is inhibited. When the lactose is high, the repressor dissociates. It permits the transcription of the LAC operon. When the glucose is high, CRP is not bound and transcription is dampened. When the glucose is low, the CMP is high and the CRP is bound. This means the operon is activated. So these are the just different words, just jumbled in different sentences, meaning the same thing, so that you can understand the regulation of LAC operon from all aspects. So that was about the LAC operon. Now let's get to the operon, which helps us understand the biosynthesis of tryptophan enzyme, amino acid in E. coli. Unlike humans, E. coli are capable of synthesizing all of the amino acids by their own cells. And the biosynthesis of certain amino acids is regulated by the operons. So, one of the such operons is the tryptophan operon. This is the diagrammatic representation of the structure of the tryptophan operon. Like the standard components of an operon, it has a regulatory region which consists of promoter, operator, and the repressor, and it consists of the regulated region which has the structural genes. In this case, all the enzymes required for the synthesis of the tryptophan amino acid. As in case of LAC operon, the trip operon has a trip repressor, which to this repressor, tryptophan goes and binds as the inducer. Now, the tryptophan itself going and becoming the inducer for the repressor means that if there is plentiful of tryptophan, or if the tryptophan is already present in the cell, the operon should be repressed. And if there is no tryptophan, then only operon should be turned on, meaning the synthesis of tryptophan should be carried on. In addition to the repression via the TRP repressor, the tripoperon has an interesting feature called attenuator. Attenuator is a small structure, small sequence present in the leader peptide of the tryptophan mRNA. This fine-tunes the repression or the synthesis of the tripoperon. When there is low tryptophan available, there should be the synthesis of tryptophan. So the complete trip mRNA is formed and all the enzymes required for the synthesis of tryptophan are available and tryptophan is synthesized. Whereas when there is plentiful of tryptophan available in the cell, attenuation takes over the role of repression of the tripoperon and a truncated mRNA is formed which does not contain any of the structural genes. I hope by this time you understand the whole idea of having an operon which consists of multiple number of structural genes which are involved in a same pathway. So if the end product, in this case tryptophan, is required, all the enzymes required for the synthesis of tryptophan will be required. If the end product is not required, none of the enzymes will be required. So the cell saves itself of the energy of regulating all the enzymes of that particular pathway being regulated by different methods. So they will be simultaneously regulated, either turned on or turned off, because they are either required together or not required at all. Before we get into the details of the regulation mechanism of the tripoperon, let's discuss a few points. So we now know that TRIP itself acts as a repressor for the operon. This means if there is tryptophan available in the cell, it should not be synthesized and hence its mRNA, the polycystonic mRNA coding for the structural enzymes which will lead to the synthesis of tryptophan should not be formed, thus blocking the transcription of the TRIP mRNA. The regulation is further fine-tuned by a mechanism called attenuation. What is attenuation? Attenuation is simply reduction of force of something or amplitude of a signal. So how does attenuation work? In attenuation, there is the formation of a hairpin structure in the tryptophan mRNA. This hairpin structure obstructs the movement of the ribosome and stalls the transcription. And since there is the translation going on side by side in case of the tryptophan operon, the stalling of the transcription further leads to the stalling of the translation, and both of the processes are prematurely terminated. So the key here in tryptophan operon is the close coordination of transcription and translation. Uh, how does attenuator actually work? So attenuator sequence, if you remember, is located in the leader sequence of the mRNA of the tripoperon. If you remember from the first slide of the where we discussed the structure of tripoperon. So in this structure, further, this diagram helps you understand various other components involved. So through until this 162nd residue, the nucleotide, it is the end of the leader peptide. You can see the there are 4 segments of the leader peptide colored and marked in violet or violet, and in red marked as 2, in yellow marked as 3, and in blue marked as 4. The important point here to notice is that the leader peptide has 2 trip codons for 2 trip residues. Then there is a region 2 which is sequentially complementary to region 3. and region 3 in addition to region 2 is also sequentially complementary to region 4. Complementarity of course means that these two sequences can bind to each other. So now this is our key, that if these two sequences are complementary to each other, they can form a hairpin structure. So which hairpin structure will form when? That is, whether region 2 and 3 will bind and form a hairpin structure, or region 3 and 4 will bind and form a hairpin structure, will be decided according to the situation of the cell, and hence further will decide whether the transcription will go on or not. So these are the regions, 4 regions, which are involved in the attenuation of the tryptophan operon. Now the first scenario is when there is plentiful of tryptophan available in the cell. When there is plentiful of tryptophan, ideally there should be the trypoperon should not be functional. It should not be synthesizing the proteins required for the synthesis of tryptophan, and so it should be stopped. How to do that? If you remember, the first region of the leader sequence of the trip mRNA contains 2 tripcotones. So the synthesis of the tryptophan protein requires the availability of charged tryptophan tRNAs. When tryptophan is in the cell, the charged tryptophan tRNAs will be available in plenty and will be incorporated in the cell, in the protein, and hence the translation will go on at a good pace. So this means that it will quickly be able to occupy the region 2. Now you remember that region 2 is complementary to region 3. But since 2 is not available for 3, 3 is now free to bind to the region 4, and it forms the attenuator structure. Now this is the attenuator structure which happens to block the transcription. It ensures that the RNA polymerase is removed and the ribosome is also removed, and hence the transcription can not go on. All right. In second case, when there is the tryptophan is present in low quantities. When the tryptophan is present in low quantities, essentially the cell requires the amino acid and it has to be synthesized. In that case, the ribosome will stall at the region 1, waiting for the charged tryptophan tRNA to be available. Now 2 will be free to bind with the region 3, form a hairpin structure. Now this hairpin structure does not form the attenuator structure. It does not obstruct the binding of the ribosome, or it does not obstruct the RNA polymerase, and hence the transcription and translation go on, and a full complete mRNA for the tryptophan synthesis of the trypoperon is formed. To sum up, we need to appreciate the significance of the attenuation. Attenuation fine-tunes the enzyme concentrations of the tryptophan operon to the range of 700-fold. This is a massive amount of fine-tuning of the regulation. This means that slightest of the higher or lower concentrations of the tryptophan should be able to regulate the tryptophan operon. There are other examples of other amino acids as well, which are regulated by the mechanism of attenuation. These are phenylalanine, histidine, and leucine. These are the sources I have used for preparing this lecture. You can go through these in case you need detailed information. And with this, I thank you for listening to this lecture. I hope I was able to convey the basics and make you able to understand the basics of both the two types of regulations in the operons. In the next lecture, when we meet, until then, bye-bye.